Yes, it's good. You see like my actual slide, not the preview. Okay, great. Awesome. I'm just going to set a timer so I don't go too wild. Okay, well, thank you so much for having me be here. It's such a pleasure to have the opportunity to talk to you all, a class of marine microbial folks. I am so excited to be here. And today I'm going to be talking about how aquatic particles are diverse and productive communities within aquatic ecosystems. And as Yannick said, I'm an assistant professor in microbiology at Cornell, and I actually just started my lab earlier this year. So since this is a marine course, I wanted to spend a moment, as Yannick had mentioned, that I'm from the Great Lakes. And so, oh no, let's see, there we go. So I want to take a moment to look at the partitioning of Earth's waters. As you are focused on marine systems, we can very easily tell most of Earth's water is bound up with salt in the oceans. Some is present on the poles within ice and a little bit buried within the groundwater beneath the ground. But the tiny bits of pieces of water that's left on the planet is found here in inland waters, and also within water vapor and rivers. So I'm really interested in this particular component of Earth's water, which is less than 0.01% of the water on Earth. However, this water plays a disproportionate role in supporting the human and animal communities on land. And so much of my work is focused on these waters. And I'm from this amazing region, the Great Lakes, and the Great Lakes has 1/5 or 21% of the world's surface freshwater. This is 84% of North America's freshwater in this single region. And today I'm talking to you from Ithaca, which is this red star over here. And if this region was to be on its own, first, drinking water here, it supports 30 million people across US and Canada. There's also 3,500 species of plants and animals that are supported by this ecosystem. And if it was separated from the US and Canada and a country on its own, it would support a $6 trillion economy and would be the third largest economy in the world. So this region is extremely important. And we know that microbes play critical roles in these freshwater ecosystems. And starting back in the 1940s, we can see this food web depiction from Lindemann, and right in the center with tons of arrows going mostly to it and also from it, we see bacteria. We know now that bacterial biomass and it fuels the trophic cascades within food webs via zooplankton grazing. We know that bacteria and archaea can be principal degraders and mineralizers of organic compounds to their inorganic constituents, like CO2. And they have major impacts on elemental fluxes. So just taking into account the terrestrial carbon that flows into freshwater systems and fuels bacterial production, that rivals the net uptake of CO2 in the oceans. So even though they're less than 0.01%, there's a lot of action going on in these systems. And they have a major influence on water quality. In the Great Lakes, we struggle with harmful cyanobacterial blooms. We also know that there's major deoxygenation events occurring, and they even mobilize mercury into neurotoxic forms, allowing it to bioaccumulate in fish that then can influence people's health when we eat the fish. So freshwater systems are valuable and they're vulnerable. And we know that today, 4 billion people, so 2/3 of the global population, they're already experiencing severe water scarcity during at least 1 month out of the year. That means that we really depend on these surface freshwater ecosystems, and due to climate change, they're really vulnerable to major ecosystem shifts. And some of the ways in which they're changing include eutrophication, which is fueled by agricultural runoff. Also, as I mentioned, cyanobacterial blooms. we're having more storms and more severe storms. And that's increasing the connection between the land and water in these systems and increasing the amount of terrestrial inputs. There's also microplastics making it through our wastewater treatment facilities into these systems, creating a new niche and new dynamics in microbes. And finally, in the Great Lakes, we have a massive problem with species invasions. And in fact, people have referred to the Great Lakes as experiencing invasional meltdown, where we just have one species invasion after another. And one thing that all of these global change drivers, the way people influence these systems, they all influence the type and the amount of particles in these ecosystems. Mostly we're increasing the amount of particles, but sometimes, like here in the case of the species invasions, the dracynid mussels, the zebra and quagga mussels, they're actually removing particles and shunting all of the nutrients down into the benthic zone, completely modifying these systems. So particles are an important system that microbes can inhabit. And there's been some really cool research finding some pieces that indicate that they're important. So we know that particles have organic carbon that's more bioavailable. And people have shown that there's a wider spectrum of enzymatic activities. So this cute little reddish orangish microbe can sniff the water, find a particle attached to it. They could excrete enzymes, providing the low molecular weight organic matter that they can take in. They could either detach and go look for more, live freely in the water, or they can die off. So there's this dynamic system that these particles create within these aquatic ecosystems. People have shown in oxygen minimum zones in the ocean that nitrate reduction to nitrite only takes place on particles. So that might halt if you remove the particles from these oxygen minimum zones. People have also shown that when you take novel particles that have been uncolonized by bacteria and expose them to marine environments, that there's these clear successional dynamics that occur. So each line in this plot over here on the right, the white indicates each line is a microbe. So down each row. And then when it's white, that means that it's really abundant. So we can see that there's a couple of taxa really abundant early on, but then they are replaced later on in time with different taxa. And that this is reproducible even across different types of carbon substrates. Some interesting work has also shown that lake methyl mercury, so the neurotoxic form of mercury, which microbes mobilize, they are 10 times higher in particles than in the benthos. And so this was, I think, Lake Geneva, where they showed that methyl mercury is much higher in the blue in setting particles than in the sediments, which was the historical perspective of where most mercury methylation occurs. Finally, some really interesting stuff, I think. People have shown that on microplastics, there's actually increased gene exchange. And so here pictured on the right in blue, microplastic from lake incubation, we can see that there's higher plasmid uptake frequencies that are 2 orders of magnitude higher in plastic than in the free living. And so there's these unique things that are happening on particles that do not happen when bacteria live as plankton in the water. And so just to kind of wrap our heads around this, I want to take a view into a micrograph from a system that I've studied. This is a micrograph from Muskegon Lake. And here in the middle, you can see this beautiful penny diatom with microbes attached to it. And you can also see some specks in the background. So those specks could be viruses or perhaps outer membrane vesicles. These larger specks are the free-living bacterial plankton. And then those that live attached with these cohorts of cells are particle associated. And so here you can see one is with this one cohort is associated with the diatom, but this other cohort of cells I'm not being able to bring up my cursor, but if you look at the other one, there's a cohort of cells that are aggregated together. And so I'm really interested in particles as microbial meeting points that can create different dynamics than those microbes that live freely on their own in the water. And I'm specifically interested in how these microbes are influenced by ecology and evolution. And this is because in order to protect and understand the potential impacts of global change on freshwater ecosystems, we need to examine the role that these particles play on microbial communities and their processes. So now I'm going to kind of transition into the goals for today. And I'm hoping that we can gain a better understanding of how particle-associated and free-living microhabitats can influence various aquatic microbial parameters. So I'm going to do something a little different. I'm just going to kind of walk you through a couple pieces and pull out 1 or 2, maybe 3 plots that kind of show different aspects of these processes. First, I'm going to pull out some data where I evaluated the community composition, looking at the particle and free-living organisms. Then I'm going to ask questions related to the diversity and the activity, specifically heterotrophic production. Then I'll briefly evaluate dormancy of particle and free-living bacteria. And finally, I'll take a view into how microbial genomes, genomes in quotes because it's from metagenomic data, and how they're tinkered by these microhabitats. But first, before I delve into these different data showing these different aspects of microbial communities, I want to show briefly how these communities are sampled. So how do we do it? So it's maybe a little bit more simple than you might be thinking, but the way that I take, which is not the only way, there are a couple other ways to sample these types of habitats. And so I use an inline filtration with a peristaltic pump where I sample the lake water. And then I mostly use a 3-micron filter because that kind of mimics the Tara Oceans data. And then sequentially filter that onto a 0.22 micron filter. Some people who study particles also might draw the cutoff a little larger, around 5 micron, or they might allow they might gather some water and allow the particles to sink and then focus on those sinking particles. But this is how I did it here. And what that looks like a little bit more clearly is that I have my water sample here, which is in this bottle labeled AB bottom. And behind that bottle is a peristaltic pump. And that follows the line down to the whole fraction. So typically the way where it says whole, that's typically the way that we've sampled bacterial plankton in the past, where we just kind of gather the sample and then filter it onto a 0.22 micron filter. But then the peristaltic pump on the right of the bottle, it goes down and you can see there's 2 filters this time. One is pointing to the particle associated filter, which is that 3-micron filter. And the second one is the free living filter, which is 0.22 microns. So these look a little different from one another. And so on the right, you can see there's a bunch of little specks. It also has a tendency to be pretty green. So there's a lot of phytoplankton or autochthonous derived carbon in this particle fraction. But in the free living fraction, it's a little bit of a light brown, and the cells just kind of form some aggregated color together. So I froze these in the field, or froze them as soon as I could. And then I performed high throughput sequencing of the 16S gene, or I use this DNA for metagenomics. And so this is kind of fueling the types of data that I'm going to show you results from through the rest of my talk. So let's take into account the composition. So I'm going to zoom into a set of lakes. It's not the Great Lakes, so it's in the Great Lakes region. And these 3 lakes, or 11 lakes, have 3 different types of productivity statuses. The oligotrophic, meso, and eutrophic. And I went during summertime and sampled the surface and the bottom during times when these lakes were stratified. And I'm going to use this data set from a couple of years ago to show you how the compositions differ, because some of these lakes had anoxic bottom waters. And so it really helps to show the magnitude of change due to microhabitats. So I'm not sure if you've covered ordination techniques, but perhaps you've seen them in another talk. This is a principal coordinate analysis. I fueled it with the Bray-Curtis dissimilarity metric, which takes into account species composition and their abundances. So if 2 points are close together in space, that means that they have similar dominant species. However, if they're quite distant in space, then they likely have different dominant species. So this is an abundance weighted metric. And what I found through these 11 lakes is that particle and free living communities are very different. If you look at the first axis, those triangles all the way over to the right, those represent the anoxic hypolimnion. So that is the lake layer, and that's around 20% of the variation of the data. We know oxygen and pH have a massive influence on bacterial communities. Second, we see the difference on the second axis between the red points, which are particles, and the yellow points, which are the free living. And this explains 13% of the variation of the data. And this is more than the nutrient status of the lake, which we typically think as being a massive influence on microbial communities. So that is quite significant. And another way to put this, which makes sense to me, but it doesn't make sense to others. So on the next slide, I'll explain it in a way that might help those who might be confused. So for here, this is indicating that particle communities are more similar to particle communities collected from a completely different lake than to the free living that co-occur in the same parcel of water. So this is a massive influence on the composition. And so particles have a stronger influence on community composition than the amount of nutrients. And free and particle communities better resemble their microhabitats in other lakes compared to each other from the same lake. So it's a massive influence on the community composition. So now I'm going to take you into a brief story about the diversity and the activity of these two microhabitats. I'll be zooming into a different part of Michigan, but it's not too far away. And this is a system that drains around 3,000 miles of land, and it's the second largest watershed in Michigan. And this flows into a very productive lake, which I then sampled for this data. And so one of the things I was wondering was the total number of counts. And so this is from microscopy counts. A lovely technician in a collaborator's lab did a lot of microscopy for us. And what she found was that the total number of cells are 10 times more abundant in the free living fraction. So when we sample communities in aquatic systems, it's more likely that the cell is a free living organism than attached to particulate matter, because they're 10 times more abundant. But if we take into account the amount of production, and here I have the community level production, and this is heterotrophic production measured through a tritiated leucine or radial labeled amino acid uptake analysis. And what we found was that free living communities are more productive overall. But if we take these two numbers and try to gain an understanding of the per capita production, so looking at the per cell rates of production, what we see is that bacteria on particles have higher per capita production. So even though they might be overall more lowly abundant, per cell, they are doing a lot more compared to their free living counterparts. So you may be wondering from the composition slide, are they more diverse? Are these more productive, per cell productive communities more diverse? And so now I'm going to show you the OTU richness. And they are. So there's more unique taxa in particle-associated communities than in the free-living environments. But when you start weighting the abundances heavily, this difference goes away. And one of the major things I was interested in was related to biodiversity and ecosystem function. And so here on the x-axis is the OTU richness that I showed here on the left. And then on the y-axis is the community production that I showed on the previous slide. And then asking whether or not there's a relationship between the two of them that fits within the broader theory of biodiversity ecosystem function in ecology. And so what I found is that only particle-associated bacteria have these biodiversity ecosystem functions. And actually it strengthens with evenness. And so this is indicating if we lose species in these particles that they will become less productive. And so that's something that we need to keep in mind when we think about global change and its influence on these habitats. Okay, so particles have fewer cells, but they're more diverse and they're disproportionately active. And only particle communities display biodiversity ecosystem function relationships. So if we lose species in this habitat, perhaps due to climate change or human influences, the productivity will go down. So now I'd like to briefly evaluate dormancy, and we're going to go away from these inland waters and into Lake Michigan. And so the lake I've been showing you is Muskegon Lake, which is here on the right with some of those orange sampling points that I showed you from the previous work. And then now I'm going to show you some samples collected from near shore and then a little further away and then out towards the center of Lake Michigan. And I'm going to evaluate both the cell number and then the proportion of dormant taxa, just to get a glimpse of, okay, even if they're more active, is this because there's a few populations that are driving that activity, or because of high dormancy, or are they mostly active? And so here on the left, I have the different stations. So on the right, it will be Muskegon Lake, and then we'll drive further out as we go left into Lake Michigan. And so what I found is that Muskegon has much higher cell abundances, and this is counts via flow cytometry. And as you go farther away from the shoreline in Lake Michigan, the cell numbers decrease dramatically. And if we take the analyses from the high throughput sequencing data and we look at which ones are more abundant in the RNA versus the DNA, then we can get a sense of each of the OTUs, or in this case, I was making ASVs, amplicon sequence variants, but we can kind of think of them as the same. And here I'm going to show you the proportion of dormant ASVs or taxa. And so we can see that even though there's many more cells on the left plot in Muskegon Lake, which is depicted as MLB in red, we can see that there's a huge amount of variability in the amount of dormancy within Muskegon Lake. But that as we go out into Lake Michigan, there seems to be a much more stable and less variable amount of dormancy. But all of this dormancy is above 50%. So most of these ASVs or OTUs within these systems are dormant. But I thought this was kind of a cool story. So now I want to take a moment and zoom back into these microhabitats and view how genomes, and here I'm using genomes in quotes because they're created from metagenomes, and how these genomes are tinkered by these two microhabitats. And I want to start with this plot, which shows that bacterial genome size is influenced by lifestyle. And this is one of my favorite plots in microbiology because not only does it show this beautiful, almost perfect correlation between genome size and the number of protein coding genes, but it also shows some fundamentals. So this is because bacteria have a deletional bias, and so they have very little non-coding DNA, very little intergenic spacers, and they have this beautiful display of their genes in organizational packets as operons. And this is all related to this mutational bias for deletions. And that's the tendency to delete DNA that's not used. And so we know that obligate symbionts have the tiniest genomes with the fewest genes. Pathogens are a little bit larger with a few more genes, and then non-host associated microbes have the largest and most protein coding genes. But how did this happen? So there are two paths to genome reduction. And one of those paths is what symbionts, parasites, and commensals take. And that path is through genetic drift. They tend to have low effective population sizes, which allows drift to be very impactful. And what happens is they end up gaining mutations and getting a lot of pseudogenes, and that reduces their core metabolic repertoire and their overall gene content. Then they might try to rescue themselves by duplicating genes, which will likely get pseudogenized again. The other path to genome reduction is streamlined free-living cells, and that's through natural selection due to nutrient limitation. This is because there's very high effective population sizes, so selection acts very efficiently, and they tend to not have fewer than 1,000 genes. There's a stable core metabolic repertoire, so they can survive on their own in the environment. And pseudogenes are rare because they would get deleted because purifying selection can be very efficient. And that leads to very few intergenic spacers. These two pathways, as we've discussed in a previous paper, are related to genome erosion when drift is really strong, or genome streamlining when natural selection is tinkering genomes to be tiny. Streamlined free-living cells have been found all over the world in aquatic ecosystems, and abundant marine bacterial plankton have been the textbook example of this. So if we go back to this plot, I'm going to hypothesize, which has already been shown, but that aquatic free-living bacteria have smaller genomes, so that hypothesis is not quite as exciting. But what's more exciting is that aquatic particle-associated bacteria might have larger genomes. So I got this particular data from Muskegon Lake, and through the channel is Lake Michigan. And so I sampled these two locations over a couple of years and a couple of months in the summertime. And I took these two fractions as I showed you before, and then I did Illumina shotgun sequencing. I assembled them into contiguous sequences. I performed binning based on the tetranucleotide sequences and the relative abundance to form population level genome bins. And this is also referred to as metagenome assembled genomes, or MAGs. This is what I was referring to as genomes in quotes earlier. And then we need to clean up our data by dereplicating and refining them. We might classify into their taxonomic groups, annotate their genes, and then think about their genome characteristics. And of course, I need to do some stats to identify the particle, free-living, and those that generalize. So I found that habitat specialization is associated with phylogeny. This is consistent with a lot of the previous work, where actinobacteria tend to be free-living, planktomycetes tend to be particle-associated, and then Bacteroidota tend to be generalists. So, does habitat specialization influence the estimated genome size? I was curious if there were signatures of genome streamlining in the free-living. And so here on the y-axis is the estimated genome size from the metagenomic data. And what I found was that particle MAGs are 2 times larger. Free-living tend to be around 2 megabase pairs, and then particles tend to be around 4.6 megabase pairs, albeit with lots of variation. And then there are the generalists in the middle. But since genome streamlining occurs due to nutrient limitation, I was curious if we could see some signatures looking at the nutrient requirements that are required by the genes. So I wanted to know if there was higher nitrogen content required by the genes in these 3 different microhabitats. So I'm going to show you the average carbon to nitrogen ratio per amino acid. So using the protein coding genes. And so if the value is higher, that means that the amino acid in the genes encoded by that genome requires less nitrogen. But if it's lower, then it requires more nitrogen. So it's a little bit of the opposite. What I saw is that free-living mags require less nitrogen than particle-associated ones for their functionings. Again, there's a lot of variation, but perhaps this could be a potential adaptation to the lower nutrients. And the generalists are all over the place. And one other thing that I will show you before I end my talk is I was curious because to be particle associated and to sniff the water, find your particle patch and attach to it and perform a bunch of metabolic functions to try to metabolize those particle nutrients and dissolve them, you really need to know and be able to respond to the environment quickly. So one aspect of that are regulatory genes. And one of the major regulatory genes that people look at are these things called sigma factors. And these are proteins that are needed for transcription initiation. And they allow for the binding of RNA polymerase to these gene promoters. So the more sigma factors that are present in the genome, the faster a bacterial cell can turn on and off genes. So I was curious if we could see a signature of these regulatory genes within the metagenome assembled genomes in this data set. And so now I'm going to show you the density of the sigma factors per megabase pair. And particle mags do have a higher density of them. Perhaps this allows them to more quickly adapt and modify their transcription to environmental changes. And then the generalists were more in the middle. So the type and habitat of a bacterial taxon has a strong influence on its genome contents. And particle mags are 2 times larger than free-living mags, and they have more regulatory genes to respond to that environmental change. Okay, so with that, I will end my talk and mention that my lab is growing. And so I'm not sure if y'all are graduate students or undergrads, but I am interested in hiring a postdoc. So if you're interested in any of this type of work, please get in touch with me. And I am here today because of a whole community of scientists who have supported me. I've had an incredible amount of people and classes that I've taken. And these are some of the people pictured here, including my committee over here on the right. Vincent Deneff was my PhD advisor and Howard Ockman was my postdoc advisor. And I've had incredible support from various funding agencies over the years that have made this work that I presented to you possible. And so with that, I will take any questions.